Hi, Maria Grazia.
Hello,
Really
hello.
Hello.
Nice to see you again. I hope this time the Wi-Fi in the UK, in Cambridge, it works.
It's looking very good. And you've clearly brought some Italian weather back with you.
I mean, yes, it's exactly like in Italy, maybe also even warmer than some parts of Italy. But it's nice.
It's lovely.
Yeah, but next week will change, apparently.
I think we all need a bit of change.
Yeah, mainly the grass is so, the garden is so yellow and dry.
It is.
Yes.
It's like the savannah, isn't it?
Exactly. When you walk, makes this noise. That
Yes.
is very strange. Yeah.
Well, we have a super group for a cuppa and cake, which is really great. There's some new faces I see around the place. Welcome to you all. Hello, Martin. And Martin.
Hello.
Good to see you all.
Thank you for the invitation.
You're very welcome. Very welcome. Alison, it's good to see you. Thank you.
Hello, thanks for having me back again.
Not at all. Not at all. I think we're going to be delighted by the introduction you've given us to Maria. Can I just give a couple of notices before we start? Our next session at the David Rayner Centre is Dr. Matthew Smith, who's an ENT consultant at Adam Brooks, and he's about to conduct some research on swallowing, which would of course be of interest to most people with Parkinson's, because it is one of the issues that eventually sort of catches up with people. So he's looking to do some research and he's wanting to seek comments from people. So I think that's going to be a very positive conversation. And probably, I suspect I'm going to spoil Caroline's thunder here. A final call for our 40th anniversary celebration at the Fitzroy
William
on the 23rd. And pardon?
Fitzwilliam.
What did I say?
Roy.
Fitzroy. Fitzwilliam, sorry. That's because I wrote Fitzroy Street in the eulogy just a little while ago at the Fitzwilliam. I suspect the ticket numbers are in excess of 150 now, Caroline.
Yes, I think, can I just say that when he talks about Fitzwilliam, he doesn't mean the museum, he means the college auditorium.
Yeah.
And Paul Mayhew Archer's Incurable Optimist. We actually have 180 bums on seats now. Some of them will be wheelchairs, so that will ease up space, but I would like to sell another 20 if possible. So if you know of somebody who might come along, then please encourage them.
Or if you've not got your tickets yet.
Oh, and also, can I add to the 26th that I'm making an anniversary cake for our meeting.
Are you? Oh, lovely.
Those who can't make it on the 23rd can have a slice of cake on the 26th.
Wonderful.
Thank you.
But it cannot be in Zoom.
Sorry?
Then it cannot be in Zoom. You have to have a real meeting.
Right, a real meeting. Oh yeah,
yes, a real meeting,
real cake,
real meeting, not cuppa and cake.
Keith, yes. Can I just add that we're on the radio on Sunday apparently?
Oh, excellent.
And there's something in Radio Cambridge as well now.
And Radio Cambridge as
Sunday, well. And Radio Cambridge another time. I'm sorry, I can't drag that up. I will try and see if I can find that out and let you know.
So I think we need a marquee on the lawn now. For another 500.
Well,
I don't know. I think never mind. I'll shut up now and see if I can find out when the other one is.
I think it's magnificent. When we started talking about this, we thought if we get 100 people, we'd be pretty pleased. It might just about cover our costs, but to be edging towards 200 is almost double our expectations. So I think it's a fantastic job, Caroline. Brilliant job. Well, as we've got our speaker with us, if I organise screen share, if I can, as we've got Maria Grazia with us, and we look to have got a good connection, can I hand over to you, Maria?
Yes.
And
okay. So thank you very much for the invitation. It's really a pleasure. Also in Cambridge, I really am very, very grateful. So please, this can be a bit of a scientific talk. So can you please stop me if there is anything that is not clear? And I will try to explain better. What I would like to talk about is the work that we are doing, trying to identify new treatments for Parkinson and try to understand the causes of the disease. But I would like also to give you some history, how we got there, and what we found during this year of research. So this, you probably know, is the old LMB, and my lab is this one here in the old building. And what we are working, we work
Clinical Neurosciences in Cambridge.
Yes.
Books.
Yes. No, this is one. Clinical Neurosciences does not have their own building. So we are spread all over Addenbrooke's. Some people are in the 4V sites. We are in the Clifford Aldo building. Some people are in the Cambridge Research Institute. So it doesn't exist. We are looking for money to create a building where to be all together, but for the moment we don't have one. But I spent 20 years almost in the Brain Repair Center. I don't know if any of you knows the Brain Repair Center, and they collaborate a lot with Roger Barker there that maybe some of you have met. So what I just get the screen. So this maybe you know, but I would like to say the history of Parkinson. Parkinson is a disease is not new. It has existed for many thousands of years. And in the Ayurvedic medicine, it was known as Kampavata. And it was a disorder that is described exactly with movement alteration problems. And it was treated with a plant that is known as Mucuna pruriens, that you can see here. And then these plants are like beans. And in regions, for example, of South America, like Colombia or Chile or other parts, where they do not have L-DOPA, they still use this plant for inhibiting the tremor and so on. And so just that you get very little compound out of the beans, and so one has to make them into powder and then they boil them in water and then drink the water. So people that I know, for example, in Colombia, they drink these 7 or 8 times a day at least to have a sufficient amount of L-DOPA. However, the description of symptoms like Parkinson are also present in the Chinese medicine, and it's called like Suwen. And then in the Old Testament, in New Testament, and finally in 175 after Christ, we had Galeno mentioning a shaking palsy. And so you see that also Leonardo da Vinci described this tremor, which was uncontrollable by the soul. So the person did not have a possibility to stop it. And then finally, James Parkinson, who was a neurologist in a village near London, described the disease in this little book that is called the shaking palsy. But Charcot in France determined that sometimes it's not the tremor, the symptoms, but can be also slowiness in the movement, and therefore he decided to call the disease la maladie de Parkinson, so Parkinson disease, as we in England. So Parkinson, therefore, you know, has always existed, but it is now very, very common because the diagnosis is much better, but in particular because we live much longer. And so people develop this usually over time, and mainly around the age of 57, 60, there is a large percentage of people that develop the disease. And these, as you know, are the characteristic feature, the motor feature, like rigidity, resting tremor, bradykinesia, and the postural instability, but they do not appear suddenly. They are not the only feature of the disease. And in fact, they appear only when you can see here there is this is a human brain. This is the area known as substantia nigra, because the cells in this area, they contain a pigment that is melanin, so they appear black. And when 50% of these cells are are dead, then the symptoms start to appear. So it is a long process that becomes evident only when these well-known symptoms start to manifest, and then people they go to see the neurologist. But the actual process starts much earlier. And what is left in the neurons of these, in the brain, and so this is the pallor, so-called, of the substantia nigra, when these neurons contain the melanin shown here, when they disappear, this is how the same brain area looks like. And then there are these sort of inclusions that here are colored with a pink dye, and that's how they look pink. Per se, they're not pink. These are all colors that we give, we use to recognize them. And so these inclusions are known as the Levy body. And this is the gentleman that described them, Friedrich Levy, who described this inclusion. You can see they can have a different conformation, but in the substantia nigra, usually they have this round shape, and described them in 1912. So because these inclusions are found in all sporadic cases, so in all the majority of cases of Parkinson disease, it means that now to have the diagnosis, the neuropathological diagnosis of Parkinson disease, one requirement is the presence of these inclusions in the brain when the neuropathologist looks at the brain. But then if one uses the electron microscope, you can see much more in depth. And this is what you see. This round shape actually is formed by many filaments. You can see here. And they are much more condensed in the center, and then they're loose in the periphery. And so this is called the halo of the Lewy body, and this is the core, also called, of the Lewy body. But nobody knew what these filaments were made of. And this it was really a problem, because many protein, if you look at this cell here, if you think you have these aggregates, these tango sort in this part of the cells, it will stop everything that moves, everything that traffic. And so you can find a lot of protein trapped in this Levy body, but nobody knew what these filaments were made of. And so when I was working in the Laboratory of Molecular Biology in the MRC with Michel Godert and Ross Jakes, we were working on another disease in Alzheimer, but we were raising antibodies that are marker for proteins in the brain. And we got an antibody that recognized two proteins that we didn't know what they were, and they were not the one that we were looking for. So Ross decided to call this protein perfectin, because any experiment you were doing with it, it was working. And then the other, imperfectin, because it was not as good as the first one. But what they had in common was that they were very similar. You see all these parts in black? Those are similar between the two proteins. And these letters are the amino acids, which are the building blocks of the protein. So they had a lot of amino acids, the sequence that was similar. And that was really interesting for us. And what we looked in the computer, in the web, was if a similar protein would have ever been described. And we found that indeed a protein known as a synuclein had been described in the electric fish. So not in humans, but in the electric fish. And these people in Stanford called this protein synuclein because they found in the center of the cells as well as in the terminal. So we decided to keep the name, and we call these two proteins alpha and beta synuclein, as shown here. So the perfectin would become alpha and the imperfectin beta synuclein. And this is how the structure of this protein looks like. So you see these square, alternated square, yellow and white. They are similar in the two proteins, and they differ in the terminal part here, one in blue and one in violet. This square, alternate square, is shown here as the amino acid KTKGV, are known to bind to lipid membranes. And this is because the sequence is very characteristic of protein, is the general sequence to bind to lipids and to membrane. And in both cases, you can see that the number can change of these repeats, but the function remains the same, indicating that this is one of the main functions of alpha-synuclein. And so if you look, this is a neuron, so a nerve cell, and this is the cell body, so-called, and this is the nucleus where the DNA is located in the neurons. And these are known, this process is known as dendrites. Then there is a very long process that transmits information through release of small molecule to other neurons or other organs, and this is called axon. And we could see that in this axon, for example, here are the terminal again, and this is how they connect. And when the neurons connect, they form these little synapses, so-called here, that in the microscope, between cells, between neurons, you can see they appear like little dots. And what we found for synuclein was that indeed you could find so many, you see the brown staining are the little dots that represent where alpha-synuclein is located in human brain. So this similarity indicates that the synuclein is present in the synaptic terminal. So in this area here, that if you look with the electron microscope, you will find that this is present. So we have this terminal, and that is the green one here, where you have all these little vesicles that contain, for example, dopamine. And this vesicle, at some point, they move to the edge, the synaptic cliff, so-called, and then they release the dopamine that is taken up by this other terminal, and then these neurons get the information from the previous one. So what happened in Parkinson, that there is no release anymore, this protein dopamine cannot be released anymore, or the vesicles are not released. What we found is that the synuclein is present in this part of the neuron, nerve cells, and what it does contributes to the movement of these little vesicles by binding to the lipid membranes that surrounds them and helps them to go to the surface of the synapse where the dopamine release occurs. So schematically, so you see that there are all these vesicles, and then this snare, so-called snare complex, that of which synuclein also is part, takes the vesicles to the membrane to release the dopamine. And this is the whole process. So the vesicles go to the membrane and then slowly release the dopamine, and then they are taken back, recycled to release more dopamine. So we know that this is one of the functions, probably the most important release of these vesicles of alpha-synuclein. But when we found alpha-synuclein, we also decided to look where it was in our genome, in our genes, where it is, in which chromosome it is present. And so we found that it is present in chromosome 4 and in chromosome, while beta-synuclein is in chromosome 5. And the following year, this paper came up from a group in Baltimore, showing that there was a genetic defect, an hereditary form of Parkinson disease with a genetic defect exactly in the area where the alpha-synuclein is located, in chromosome 4, Q21, Q23. And the following year, the first genetic cause of familial Parkinson disease was described, and it was a mutation in the alpha-synuclein gene. So what means is that there are some people, and that this was identified in a very, very large Italo-American family known as the Contursi family, and in 4 Greek family. And this genetic mutation is a change of an amino acid. So what I have shown you before, these building blocks of the protein that are known as amino acid, one of them changes. And this change is able to cause Parkinson in the patients where this. And because it is in the DNA, it is transmitted in the family from the father, for example, to the son or to the daughter, and vice versa. And this is the first description of an hereditary form of Parkinson disease. And so this was clear, was in the synuclein that we knew. We had all the tools to investigate if synuclein aggregates could be present in these filaments that I showed you before, were forming this Levy body. And so what we did, we used these tools that we had, and indeed in August 1997, only 2 months after the mutation was described, we described these inclusions that are the Levy body for the first time seen containing alpha-synuclein. And actually, I remember that I was working at the Brain Repair Center, and I did this experiment. I had just moved to the university from the Laboratory of Molecular Biology, and this was the first cell that I saw, and it was so exciting to look in the microscope. I had never seen a Lewy body, so it was really, really beautiful to see this type of staining, but we really didn't know what it meant. Was one of the protein that was just trapped in these big aggregates, or it was really relevant? So what we need to do was to show if it was present in those filaments. And so here you can see, these are the filaments that we extracted from the brain, that this is from brain of patients with Parkinson disease, this with dementia with Lewy body, and this with multiple system atrophy. And this is because in all cases, when you stain these filaments, they had this similar pink color with the dye I showed you before. And then we try to reproduce these filaments just by taking a protein, alpha-synuclein, produced in bacteria, put in the test tube and shaking it at 37 degrees overnight, and to see if we could form filaments. And indeed, we form these filaments that are artificial, are not extracted from the brain. But the fact that they were similar, and these black dots indicate that our markers recognize all of them, indicating that really alpha-synuclein was the main component of these filaments that we had seen in Lewy body. And it was different, for example, from the filament that we had previously seen in Alzheimer. This is like a filament found in the brain of Alzheimer patient, and this instead is an alpha-synuclein filament. You can see there are no black dots that are indicating that is not recognized by our markers. And then using these antibody, these markers, we colored the brain of some patients with dementia with Lewy body, or with Parkinson, or multiple system atrophy, or pure autonomic failure. And in all these cases, this brown staining indicates that alpha-synuclein and the filaments formed by alpha-synuclein were present in these inclusions. And these indicate that alpha-synuclein not only caused the disease by genetic mutation, but also was present as aggregated in the Lewy body in patients also that did not have mutations, indicating that there was a link somehow during our life, can start to aggregate and then to form this characteristic inclusion that will lead to disease. So all these diseases are listed here, all these conditions now are also known as alpha-synucleinopathy because they all contain Lewy body or other inclusion with alpha-synuclein that are associated with the appearance of the disease. And so these, as I mentioned, these Lewy body were recognized by eosine in pink, but then the neuropathologists use ubiquitin, which is another marker, but doesn't distinguish between different protein aggregates. And instead, synuclein gave a very specific staining. So using synuclein, it was possible to recognize the Lewy body also in diseases where they were never been described. And also in diseases where they were described, the synuclein recognized 25% more inclusions, more aggregates than the ubiquitin itself. And the other thing is that, for example, this led to the identification that about 60% of cases of Alzheimer's disease, they have Lewy body in a specific area of the brain, not like in Parkinson, the substantia nigra, but in an area of the brain that is known as amygdala. And nobody knows why they are there, why they are localized in that specific area and nowhere else. So the Lewy body then, using these markers, we could find that it's not always round as described by Levy or condensed, but it has a history. And so in these nerve cells, synuclein start to form these little aggregates that you can see here as red. They become more and more until at some point in one area, they are very, very close and abundant, and a first seed of aggregation forms, like in this case. And this seed of aggregation starts to attract all the synuclein that is around, okay? And then form the first body of aggregates that is known also as a pale body. And then this pale body starts to condense and forms the core, as we said, as I showed you before, and then the halo, as we find in the substantia nigra. And this is an example of all these stages in the formation of these aggregates in the brain of a patient. You see, these are 3 Lewy body in one of these neurons, but this is not a Lewy body, it's just some accumulation of alpha-synuclein, as for example, it happens here. So we know that with these aggregates, with these markers, we could understand more about where the disease starts and where it goes. And so this gentleman, that is Heiko Brack, and his wife, Kelly Del Tredici, when they were in Frankfurt, and the previous wife of Brack, Eva Brack, who died, they start to look at thousands and thousands of people that had no disease, people that had different disease, and people that had Parkinson. And so they just look at people that were 20 years old, 25 years old, and they took various organs. So beside the brain, also the gut, the stomach, the heart, and they start to look for alpha-synuclein deposits. And what they found, that they could see the beginning of the alpha-synuclein aggregation in the gut and not in the brain. And so they come up with a theory that perhaps alpha-synuclein start to aggregate in the gut, and then from the gut, these little aggregates through the nerve terminals, they reach the brain. And then when they have reached these nerve cells in the brain, they start to spread to other nerve cells that are connected to this one. And so at the end, you have this theory that the gut could be the origin, but also Aiko found that another possibility is the nose. So the nose also could be an area where alpha-synuclein is present, and from there could aggregate and could just spread through the nerve connection to the brain. So now we have this periphery. And why the nose and the gut are important? Because if you consider, these are the two main parts of our body that are exposed to the external environment. And therefore, according to Aiko, Brack, it is possible that there are insults that lead from the external, from the exterior, they lead to alpha-synuclein aggregation. For example, could be pesticides or could be other factor, but the pesticides are the best known, that could trigger this aggregation, that could spread then to the brain. And in some people, it goes on, and in other instead, depending also on the genetic background, can be stopped. And so these aggregates can be degraded. In others, instead, they can go on and spread the various areas of the brain. And this was a theory until, as you probably know, one of the treatments that people in Sweden try to perform, and also Roger Barker here in Cambridge, is to transplant cells into the brain, substituting the dopaminergic cells that do not produce dopamine anymore. Usually these cells are taken from aborted fetuses. And therefore, when in this case, a patient had the transplant for 15 years when he died. And so it was possible to trace these cells. And it was very, very surprising that these cells contain alpha-synuclein aggregates. They were belonging to somebody that was very, very, very young, where nobody had ever seen this. So this indicated that these cells in the transplant were taking the alpha-synuclein from the surrounding area. And this was the first demonstration that alpha-synuclein could spread from one point in the brain or in the periphery to the to other nerve cells, exactly like Brack had hypothesized. And so this is what Brack suggested, that it could start in the periphery, arrive to the brain here, the brain stem, and then slowly spread, the Lewy body spread to all this area. And if they stop here, there will be Parkinson. If they go to also this other area, it could be Parkinson with dementia. And based on this, these are the symptoms that now are known to occur, that possibly they can occur before the motor symptoms, like constipation could be one, and it could happen 20 years before the motor symptoms or more. Olfactory deficit, and the one that has been connected about 10 years before the motor symptoms is REM sleep behavior disorder. This is disturbs in the sleep where the person acts their dreams, and therefore are the people sleeping with them that realize that if they become aggressive, they are dreaming something, really they are like if they were awake. And so these are also other symptoms that they could be associated with this progression of alpha-synuclein aggregates spreading. And therefore, as I mentioned, in some people, there are very few Lewy body, and they really do not have any symptoms and remains like that. In others, these aggregates spread, leading to Parkinson's disease, and in others, they lead to some form of Lewy body dementia. And according to what Brack suggested, now it has been reported that studying live patients based on the symptoms and from this group in Denmark, that the pathology could start from the brain and therefore from the nose, the alpha-synuclein, and then spread to the gut. So the gut symptoms will be the last one to appear. And in its spreading, we know that also reaches the heart. And then if it starts from the gut instead, the sleep problem will appear very soon before the motor alterations. So what we want to know after having known that alpha-synuclein is involved so closely to the progression and the disease was to produce transgenic mouse models where we can reproduce this process to test drugs that could stop it, and mainly that they could be a treatment based on the mechanism of the disease, not just a replacement of something that is missing, but something that could stop the loss of dopamine, for example. And so in this mouse model, we want to have in the striatum, so these, the cell body that the nerve cells are in the substantia nigra that is called pars compacta, and the terminals that I show you, they end up in this area that is known as the striatum. And there, these neurons release the dopamine. So what we want to have a mouse where we have this deficit, progressive deficit in dopamine release, loss of total dopamine, and then at the end, death of dopaminergic cells, nerve cells. And so we produce a transgenic mouse where all these should be associated with alpha-synuclein aggregation. So we know that it was really alpha-synuclein the cause of all these. And so what we did was, you can see this is the shape of the substantia nigra, pars compacta, and then these cells are stained for tyrosine hydroxylase, that is an enzyme involved in dopamine production. So you can see here that these cells, they all contain alpha-synuclein in these mice, and we use a mouse where there was no alpha-synuclein in the background. So this mouse did not have alpha-synuclein. And we know that removing alpha-synuclein, the mice do not die, are okay. And so we could see here, these cells start to express the transgene that we put in. And this transgene, that is alpha-synuclein, start to aggregate in the mouse neurons where there is no melanin. So you don't see the black pigment. In mice, there is no black pigment. And you see that they form these aggregates, and these aggregates over time lead to the disappearance of the processes that you can see here in mice that are 3 months old. Slowly they lose the processes, and then when they are 12 months of age, you don't see any processes anymore. And this also is evident in the striatum. So this is substantia nigra, and this is the striatum. And if you look in the striatum, all this happens at this level of the synapse, which indicates that this alpha-synuclein that form these darker dots is aggregated at this level. And so we look in the mouse staining this area, and we could find, you see these that look like rubbish. Actually, they are the beginning of the aggregates of the protein in the transgenic mice, and in the process, this alpha-synuclein recruits also other proteins that are involved, like synaptobrevin or other synapsin and so on. Then we were curious to see, is this something that happens only in the mice or can be found also in a human brain? And we went to look, and in human brain, very, very early on, in brains where the disease has been diagnosed for 2 or 3 years, you find that there are these aggregates, these little dots that are present very early on in the same area in the striatum. And this is, if you take super resolution microscopy, so a form of microscopy that allows to see protein at the level of nanometer, so it's extremely sensitive and very, very little protein, you can find that synuclein here in red in our mice that are 3 months old is quite widely distributed. And green is a marker to indicate, that we are in the right place, so at the synapse. 6 months, you start to see the first aggregates of alpha-synuclein, and then at 12 months and 18 months old mice, you can see that the amount of synuclein aggregate is very, very high. So we look in these mice, if there was any death of the neurons associated with this increase in alpha-synuclein aggregation, and we can see that the death at 6 months of age, we have the beginning somehow, but at 12 months of age, we have 30% loss of dopaminergic neurons. And then at 15 or 18 months of age, we have 50% loss of dopaminergic neurons. We want to look at the dopamine release if it was altered. And then we find that when the mice are 3 months old, and as you have seen, there are not really alpha-synuclein aggregates, what we do, we put a cannula in the brain that reach the striatum, and then we see how much dopamine is released. And in red are the transgenic mice, and in green are the healthy mice. And so what we see, there is no difference in this case at 3 months, but already at 6 months, we find that the transgenic mice, after stimulation, release less dopamine. And the amount is a further decrease at 9 months, and then at 12 months of age, when we have also lost death of the dopaminergic neurons. So we had a mouse model where we have alpha-synuclein aggregation, progressive death of dopaminergic neurons, and then loss of the terminals that release the dopamine. And so we did a behavior study, and we saw that only at 20 months of age, when 50% of the neurons are dead, the mice do not walk very well. So the defect is only at 20 months. And also with 2 different methods. This one is a mouse that has to walk in something that is rotating. Here is a mouse that has to walk in a rod. And this rod can be of different size. And so when the rod is larger, the mouse can walk properly, but when it becomes small, cannot walk properly anymore. So we use this system to see if the mouse can control its movement. And then you see that only at 15, 20 months of age, this is happening. But if we look at the dopamine release, already started 6 months. And this result was really, really important because it indicates that the loss of dopamine occurs much, much, much earlier than the death. And if we can find a way to start treatment and prevent the loss of neurons, we could also prevent the development of the disease. So we look at our mice when they walk. So you just take a platform that is transparent and you put the mice walking and you measure how they walk, like the stride length, the stance, and you see that they all had a deficit in the walk pattern compared to the controls that are in blue. And so we decided to test a compound that was produced by our collaborator in Germany. And this compound is called ANLE138B and can be given in the food. So we gave to our mice from 9 months of age to 12 months of age, the time when the loss of neurons develop, as well as the behavior, the walk that is not proper anymore, the stance and the stride. And then we went to look what was happening. And what we found that this compound was removing these aggregates of synuclein, you can see here, making it much, much more diffuse, and was rescuing the release of dopamine. You can see these are the mice treated with placebo, and these are the mice treated with the drug, the transgenic mice, and these are the control mice. So you can see that we can rescue the release of dopamine, and this is associated with prevention of neuronal death. So at we can find 12 months of age. This is the placebo mice that have lost the neurons, the nerve cells, while those treated with the drug, they are not. We again studied the behavior and the way of walking of our mice, and we found that the treatment with the drug was rescuing this defect observed still with mice treated with placebo. It was not present anymore. For example, the stride length or the stance and so on. Not everything was rescued. For example, the stance duration in the hind limb was not fully rescued, but in the forelimb, there was a rescue of all the parameters with the drug. So we tested different amount of the compound to see which one was the lowest amount that could be used to prevent the loss of dopamine, so to rescue the loss of dopamine. And this result is quite important because it shows that we can use amount much smaller than what was suggested by our collaborator in Germany to obtain the same results, and so to rescue the release of dopamine upon stimulation. And this is important because now ANLE138B is used in patients in trial, and the first trial for safety and tolerability has already been done in 2020, and it has shown to be safe. And after that, the company known as MODAG, who our collaborator set up in Germany, now they have joint effort with Teva, and the drug is in phase 2 trial in patients, and we will know the results in autumn if it has an efficacy or not, and if it can be used as a treatment for the disease. So this to us was very, very important because potentially, if the results in our mice are of any significance, this could be a really, really good drug for preventing the death of the neurons and the progression. But we need another approach to show that it is really the defect in the synapse and not the neurons and the cell body that aggregates. And so we use this, we consider this other protein that is called cysteine string protein alpha, that is present in the synapse with alpha-synuclein, and it is involved in the same process, but from a different angle. So it does not really interact with alpha-synuclein directly, but it has the same function to take the vesicles to the membrane and to lead to release of dopamine. So we knew from study done in Stanford in America many years ago that when this CSP protein alpha is absent, there is disease, development of disease. But if they gave alpha-synuclein, supplement the mice with synuclein, this defect was rescued. So we decided to do the same thing, we look in the brain of our mice, and we found, you see here, there are many, many brown dots indicating the presence of this protein CSP alpha. And this in our mice, this protein is reduced. And as shown here, this indicates the amount. You see, it's high in the control mice and low in the other mice. So we decided to give to the mice the protein CSP alpha to replace the one missing, and to see if, because it acts only at the level of the terminal of the synapse, this is showing the increase that we induce in the mice, we could rescue dopamine again. And what we could find was that indeed, if you look at this, is here, this line with the red dots, these are the transgenic mice treated with this CSP alpha protein, and we can restore the release of dopamine like observed in the control mice, that here are all the lines in blue are control mice. Instead, the placebo, that is this one, you see that, and the normal mouse, they are really very low. So still the defect in dopamine release is still present. So what this is, this like before, we find that the alpha-synuclein becomes disaggregated and redistributed the synapses after this treatment. So in conclusion, we can say that what we believe, and many other people now believe this, we have this, the vesicles containing dopamine at the level of the synapse. These are the proteins that together with alpha-synuclein help to move the vesicle from this area to the surface. And then what happens is that there is release of dopamine. But in some cases, when there are alpha-synuclein aggregates, alpha-synuclein attracts in its aggregates also the other protein. And so there is no way for the vesicle to go to the membrane and to release the dopamine. And this process, it initiates like very, very few molecules, and then it increases, recruiting all the alpha-synuclein that it is around, and then causing the disease, and retrogradely the death of the nerve cells where this starts. So we are trying to reproduce this not only in mice, but for example, these are neurons generated from a skin biopsy of patients with Parkinson. And we take the skin biopsy, a very little biopsy of 9 millimeters. We dissociate the cells that are contained in the biopsy, and then we make them like stem cells, but then we treat these stem cells in a way that they become neurons, like the one we have in the brain. And you can see here that some of these neurons start to accumulate alpha-synuclein. These are the green dots here, or the red dots here, when they are taken from patients with Parkinson disease. And these are the cells that I showed you before in the mice. These are the ones that I show you in human brain, the beginning of alpha-synuclein aggregation, which is quite similar to what we see in these cells. So we really hope to have another model where we can screen for compounds. And indeed, we are using now to these cells to screen new compounds from our collaborator in Germany, but also we collaborate with people in Finland and in the UK to test compounds that they can disassociate these very, very initial aggregates. And in the meantime, we have, however, to determine when this starts. And so we are also looking for biomarkers, and we have done some study where we can take the saliva, and from the saliva, we can predict if the person has Parkinson disease and alpha-synuclein aggregates or not. And we are trying now to collaborate with our colleague neurologist here in Cambridge to make it possible to use it for making the diagnosis. And the other thing that we are doing, we are creating, finally, I'm sorry, I'm very long, we are producing a mouse to reproduce the hypothesis of Brack, where we express alpha-synuclein, here in brown, in the gut, directly in the villi of the gut. And we have found that from the villi of the gut goes to the neurons, to the nervous system surrounding the gut, and from there it goes to the brain. And these are neurons in the brain where this gut synuclein has arrived. We are now doing this, studying these mice at different age, and we have also had for this the support of Parkinson UK, but all the previous study that I show for ANLE has been supported by Parkinson UK. We are now going to continue this study in these mice. For example, we have found that some of the alpha-synuclein can reach also the heart. So we want to know when it reaches the heart and how we can prevent that. And so in conclusion, I hope that I have shown you, and has not been too complicated, that alpha-synuclein aggregates can form in the striatum very, very early on, and they cause the dysfunction of the neurons, the death of the axon, so the process that connects the neuron, the cell body of this nerve cell to the striatum. And then at the end, when the alpha-synuclein accumulates, we have the Levy body, like in this case. So the secret now is to find the treatment to understand when the disease starts in a person, which is much before that the motor problem appears, and then we can really cure possibly this disease. And the work I have described has been done by many people over the years in the lab. And so Joana Dominguez is a PhD student from Portugal that now has gone back to Portugal. She studied the mice with spreading from the gut to the brain. Laura Calo is now working in a drug company, in a pharmaceutical company in Leiden, in Rotterdam. And she has contributed to all the transgenic mice and the stem cell work. Mikael now has his own lab at the equivalent of an academy in Warsaw in Poland, has gone back and started his own lab. George Tofaris instead is still in the UK, and is now was a PhD student of mine that made the first transgenic model, and now is professor at University of Oxford. And Pablo Garcia Reitbock also was involved in the mice, and now works at the Royal Free in London. And this work is continued now by Solene Ferreira and Ziguan Zheng as a technician in the mice where there is the spreading. And we are now starting with Teva. We have been asked to collaborate to continue the work with our mice that are a good model for the progression of the pathology for other compounds. And also the Michael J. Fox has given a large amount of money, a very big grant with people in London and in Oxford, to study how the disease progress and to identify compounds that they can stop it. And we contribute, we study in our mice. And thank you very much for listening. And please just ask, I don't know, I hope to have been clear. Thank you.
Grazia, thank you so much. I think a very complex subject, which you've explained to us lay people remarkably well. I don't know whether you want to say anything, Mark. Unmute.
Thank you. No, well, look, I'll say a few words at the end. I mean, there are a number of questions that I can see on the chat. That Alison has asked a question, and I've got a question too, if I may. So, in whichever order, Keith.
Let's do questions. Ask your question. Fire away your question first, Mark.
Thank you very much for fascinating and really good to see you again. It was a long time ago that I saw you at Addenbrooke's one time. I've been reading about AlphaFold, this Google Maps of proteins. Perhaps I'm using the wrong sort of analogy. I'm just wondering the extent to which that is really helpful for your work.
From the future, that is yes. For our work now, the people in the MRC, they have just submitted the paper with the fold of alpha-synuclein in Parkinson's disease. And the alpha fold is what it does. You put inside a protein sequence and you get the fold that is possible. So in that sense, perhaps that could be useful, for example, for PET ligand, specific for synuclein or for other things. But now we have already the cryo-EM structure that could be used for that as well. But certainly the AlphaFold will save years of work for understand this protein. It's amazing.
Yes. Yes, well, the reports have been very encouraging, but you never
absolutely.
quite know whether they've got the balance right. But good, thank you very much.
Alison, you'd asked a question. Do you wish to pose that?
Yes, thank you. I'll try to. It was just I love the diagram that you had of the dopamine release and it sort of how it kind of couldn't get into the system. And it just occurred to me, is there therefore the dopamine still there and can't get in, or is the knock-on effect that there's less dopamine around?
What the possibility is that the dopamine stays there and then becomes toxic, because per se if it becomes a lot, they create oxidative stress. And then when the neurons start to dysfunction, then the synapse degenerate. And so there is no synthesis anymore of dopamine. So when you have the aggregate stop to be synthesized. Yeah. Okay, but it is two ways, because at the beginning there is this possible accumulation that could be toxic.
Causes problems in itself.
Yeah.
Okay, fascinating.
Thank you. Deb, do you want to ask your question?
Yes, Maria, thank you very much for that fascinating talk, albeit very complicated. I was just wondering, from the research that you've been doing and all these various tests, are you going to be able to devise a test which will indicate whether someone is likely to get Parkinson's disease? And the reason I ask this is because my dad had Parkinson's disease, but his sister has also got it. And obviously I am somewhat concerned about the prospects for me and my brother with regard to that.
Yeah, so there are already ways to know if there is a familiarity. I don't know if you have seen, neurologists should have advised that, because normally if in the family more than one person has it, normally one has genetic testing if he wants to know. And now there are, Y-synuclein was the first gene that was identified where just the normal protein was this mutation, the change of one amino acid. But now we know that just in some people, you have more than what it is needed. And that increase in the expression leads to the beginning of the aggregation. And then also just the normal protein, but much more than expressed in higher amount can be toxic. So yes, there are tests that that can determine if there is any possibility, or to exclude the possibility that can be an hereditary form. And these tests, there are many genetic tests. And what we are trying to do is, what I meant with the saliva, it is to see if we can detect very early on the alpha-synuclein aggregate. But of course, we can do a screening in general population, but it is more likely that you do that in people that they show some symptoms for the moment.
Thank you.
And I mean, if I wanted to pursue that, where would I go to pursue that?
I think that you should contact the Addenbrooks.
Right.
and Roger Barker.
Okay.
Or somebody in the team in Addenbrooke's, they will
Thank
follow.
you. Thank you.
Yeah, that'll be the Brain Repair Centre, Deb.
Yes.
Right. Okay.
Pam, you've got your hand up. Unmute, Pam.
There we are. Yeah, following on from that, if there is a possibility of detecting whether somebody might develop it or is inclined to, is there any prospect of any medications or anything that could change that as of now, or is that something really for the future?
Well, they are starting now, for example, there is a lot of work going on, and also vaccine for alpha-synuclein, the aggregated form. There are many antibodies that are produced to destroy, just inject the antibody and destroy these little aggregates. And then also all these modifiers. So if we find a way to stop this aggregation, that could become a cure. But when the aggregation has already killed the cells, you don't replace the cells if you don't do a transplant. And so that is why we have to arrive quite early, at the very beginning, before there are really the symptoms. And I think that there will be something very, very soon, because there are so many drugs in trial at present. That there isn't something right now, but it should be very, very soon, because we should have results also of the treatment with antibodies or vaccine and so on.
Yeah, thank you. I just that I don't think I want to know if there's nothing I can do about it anyway.
Yes, no, I can understand that.
Good luck to you.
Yes, but for example, the important will be to prevent the nerve cells to die.
Yeah.
And that, because that is what maintains the functionality. So that will be the function of knowing.
Right, to make sure one is exercised in every way. To try to, yeah, right.
Yes.
Thank you.
Yes.
Yeah.
Any other questions, anybody? Martin.
I'd just like to thank Professor Spillantini for a really excellent talk and ask her if she would comment about 2 trials using humanized monoclonal antibodies, which have not been successful in actually alleviating symptoms in Parkinson's.
Yeah, so at the moment there is one antibody that they have not found very successful, but there are many more. And these antibody, they are all in trial. They are for different parts of the protein. So nobody knows. They have made these antibody for one part of synuclein. And now they are testing different parts. So there is still the hope that an antibody will work. But what we work on is instead to find, like Anle, to find this small molecule that can be just taken in the food and disaggregate the aggregates, and then to maintain them not aggregated. So the results of this very, very small pilot study that to which also the UK participate, I think it should be known in the autumn. And then if depends on how they are going to have much larger trials. So at the moment, there is not a miraculous drug, but there are many trials done by the Michael J. Fox. Many companies are working on this. So we hope that sooner or later something will come up.
Can I come back on that?
Sorry?
Please.
I noticed that the studies are done on removing alpha-synuclein from the blood.
Yeah.
Is that a logical procedure?
No, I think that is only a test to see if it works, because removing synuclein from the blood is not really it doesn't work. You have to remove from the brain.
Exactly.
And so the blood, I think they are doing that to see if the general approach works, because synuclein can be found also in the blood. And so if it is reduced in the blood, it means that the compound works, but they have to make sure the compound goes to the brain. That's where it has to work.
But you wouldn't expect the monoclonal antibodies to get into the CNS, would you?
No. And that is one of the main question, because normal antibodies are not considered, but what they try to do, they say to try to, because there is this spreading of the protein, they try to remove the one that goes out of the cells and then can be spread to other parts. So the antibodies seem that that is the function that people expect, to remove the one that is transmitted. However, strangely enough, somebody has also reported in a meeting recently that they have found that also the antibody could go in the neuron, which is very strange. So I don't know if to believe it or not. And the other thing is that people now, they are engineering the antibody in such a way that can cross the blood-brain barrier. So they can be like with transferrin receptor, they bind to the receptor, they add another molecule, and they are catapulted on the other side.
That's fascinating.
Yeah, the other thing is that the blood-brain barrier with age and so on becomes much more leaky. And that is really another thing that is considered, that it could be leaky and could let some of this molecule not to cross, because to let inside, to go inside. So there is quite a lot of work about this.
Thank you very much.
Mark, you've got your hand up?
Yeah, I'm just wondering about time. I've just got a quick and cheeky question for Maria, but just very quickly. I was going to ask it. It's an impossible question, but for you, is synuclein really the the cause of Parkinson? And the question now is to find therapies that can interrupt that effect, because there are so many different views and lines of research. Yes. And for you, is this it? Or is, and I know it's an impossible question.
Yeah, I know there are different questions, different answer to this. I know where you come from. And I have to say that the synuclein is for absolutely the majority of cases, because it is present in the living body, so it has to mean something that is there, but also because there are genetic mutations that are associated with disease. So for example, the first was a change of amino acids. There are not many. Now we know there are 5-6 change of amino acid that cause hereditary Parkinson disease. But what is interesting, there are also duplication and triplication of the gene. So it's not a change in amino acid, it's just the dosage. How much of this protein you have in the brain. And then when you have a triplication, you have Parkinson disease and dementia with Lewy body and so on. So if you look at other genes that we now know cause Parkinson, very often they are associated with Lewy bodies. And however, if you look at the form of Parkinsonism, that is Parkin mutations, they don't have Lewy body. So in that case, there has to be some different way to reach the end point, which is killing the neurons that are in the substantia nigra. But for the sporadic cases and the cases where you have Lewy body, I think it is critical, the synuclein aggregation. And then I know also there is a lot about mitochondria and the energy that the mitochondria is. Yes. And so it is true that there are models where you give compounds like MPTP or 6-hydroxydopamine that they kill the same neurons, but they don't lead to alpha-synuclein accumulation. but those models are acute. So do you give a dose and the following day the neurons are dead? What we are talking about is something that lasts for 30 years. And so there are a chronic model of MPTP which block the mitochondria, the complex one, and they lead to accumulation of alpha-synuclein. I think the real question is to find what is the link, because now there are reports that mitochondria under stress, they recruit alpha-synuclein. Yes. And this could be the beginning. And so the mitochondria is really what starts, but the synuclein then it's what continues in the sporadic case.
Thank you. No, that's really very helpful.
Caroline, you've got your hand up. You sent me a note, but I'm not quite sure what it means.
Hi, Caroline. She is mute, Caroline.
Yes. No, I'm sorry. Thank you so much, Dr. Spillantini.
Maria. Is it?
Maria.
Maria is.
I'm glad we were able to get the link to you. I was just going back to the really mundane issue of radio broadcasts. I have put the very first chat in the chat stream, tells you when Tony Barnfield's Cambridge 105 radio show is, and also when Radio Cambridgeshire is on Friday the 19th. So if anybody's interested in listening to those, just make a note from my chat up at the top of the box.
Okay, thank you.
That's it.
Barry, you make yours the last one.
I'd just like to ask a question with respect to the excellent talk, and that is that basically if you treat with a substance like Anli that prevents the problem of loss of dopamine release, can you treat after a period of time, and do you see any improvement in functions?
Yeah,
with
so
things like that.
That is what we are doing now in the mice. So what we have done, actually, we stopped the treatment after 3 months, and we have checked after 5 months, for 5 months, and it seems that still there is an improvement after stopping it. But of course, the mice is a mouse. And so the aggregates, they last for not forever. I mean, they are there forever, but you cannot check after 2 years because they die after 2 years. That's the lifespan, normal lifespan of the mouse. But with these trials, that is exactly what they are doing. So the trial that is ongoing now is in Parkinson cases. And so that will show if there is an improvement, and the results should come up in the autumn.
As a supplementary to that, is there any evidence or is there any work going on trying to use a different animal model other than mouse, an animal that lives for longer?
Yes, but if you look at monkeys, they can take 16 years or 15 in order to age. So it takes a bit long. Yeah. They are doing also tests in monkeys. But in the monkey, you have to, again, have this injection of alpha-synuclein fibrils, which induce the aggregation of these endogenous of the protein of the alpha-synuclein that is in the brain of the monkeys. And so they are doing this work in Bordeaux, in Arizona, and they used to do that in Paris, but they don't do it anymore. The fact is that work with monkeys is very difficult. However, that's what they are trying to do, to have a different span, because as you are right, the mice, they cannot live more than 2 years. But I think that will be really important to know the results of the trials. That should be soon. Yeah, sorry.
I come from an agricultural background originally, and I wondered about farm animals because there's a lot of interest in genetic modification of farm animals.
Yes, yes. So yes, the sheep, for example, that is something that one could study, but so far they have not. We have studied the brain of sheep or bear for different type of aggregates. but you need to know that they have the aggregates. There are no models that they reproduce the symptoms that you can detect the effect of the drug. You have to go and look at the brain.
Sure.
And so you need something where it is reproducible, the aggregation, and then you know that you can stop it. And that you can go back and check.
Grazia, I think we've taken well over an hour of your time.
Oh no, I am really happy. I really enjoy talking to you. I really enjoy. And please just contact me if you have other questions. And it's easy to find my email. You can ask Keith or Caroline.
Thank you. I feel very privileged to have listened to your speech, to your talk today. I think it's been.
I feel privileged to have spoken to you. So.
And if there's anything that you need from the branch, please don't hesitate to contact us.
Thank you very much.
It's been a wonderful talk. Mark, do you want to do a closing comment?
Yeah, just very briefly. Thank you very much, Maria. I didn't know the early history, and I didn't know about the Ayurvedic sort of remedy, and so that was fascinating. In all my time dealing with Parkinson's, I've not come across much before James Parkinson's himself. Yeah. So thank you for your your talk about what really is quite breakthrough science, and both in terms of early diagnosis and potential cure, and at the very least a high probability of being able to moderate progression and alleviate symptoms. So a really very exciting and well done, and thank you for all the work you're doing for us, and it's great to see you again. Thank you, Maria.
Thank you. It's really a privilege for me. Thank you very much.
You're very welcome. Thank you so much.
Thank you.
Thank you, everybody. I don't want to continue. I think we're leaving on such a high. Thank you so much. I'll get the recording on YouTube within the next couple of weeks. Thank you so much.
Bye-bye. Thank you.
Bye-bye.
Bye.
Bye.